Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Excel International A-Level Biology Unit 5 Scientific Article for October 2024. These are potential topics. So I will view through the article and then I'll talk about the topics that questions can potentially come from. This is subdivided into two articles, so I'm going to begin by reading the first article. We've been studying the same people for 76 years. This is what we found out about Alzheimer's disease. In 2016, a sub-study of 502 people from the cohort, known as Insight 46, was studied to specifically study brain aging and dementia and their life course influences. Using cutting-edge imaging and AI technology, we have been observing their brains ever since. The results from our studies have revealed several important insights, including: the first is cognitive function in childhood relates to cognitive performance 70 years later. The second, education does not just increase opportunities but is significantly associated with brain health in later life. Midlife appears to be the time when hypertension and cardiovascular risk may influence dementia risk. Number 4, while weight gain in midlife has many adverse health implications, weight loss in later life may in some cases be a sign of impaired brain health. Next, we go to tackling subtle variations. When members of the cohort reached their mid-30s, Mike Wadsworth, the then director of the NSHD, focused the study on physical and mental health, particularly the measures that show subtle variations in general populations such as blood pressure, lung function, physical performance, and emotional symptoms. This approach was enhanced by the subsequent directors. We have Diana, Koo, and Nish. The last name, I'll just skip it. Cognitive function was picked up again at the age 43 with a new emphasis on skills vulnerable to decline, such as memory, concentration, and mental speed. These measures, along with those of physical and psychiatric health and their life course influences, form the platform on which we built our brain study Insight 46. What is dementia? Dementia is an ancient word meaning out of mind, but today it refers to a syndrome of acquired, not present from birth, progressive cognitive impairment, severe enough to interfere with everyday activities such as planning meals, managing bills and medicines, and housekeeping. As cognitive impairment worsens, these activities become more impaired, eventually disrupting basic self-care such as dressing and bathing. Sometimes this is accompanied by depression, paranoia, aggression, wandering, and reverse sleep-wake cycles. Dementia is therefore very different from the mild cognitive changes that occur in normal aging. Alzheimer's disease is the commonest form of dementia. On a biological level, a key process is the depositing of beta-amyloid protein in the brain. This is a protein that comes from the fatty membrane surrounding the nerve cells. It is chemically sticky, gradually clumping together and interfering with nerve function and triggering inflammation. The clumps gradually gather between the nerve cells in the brain of people with Alzheimer's disease and become plaques, hard insoluble accumulations of beta-amyloid proteins. These plaques are thought to be an early hallmark microscopic feature of Alzheimer's. However, amyloid plaques in themselves are not sufficient to cause dementia, which is more closely related to accumulation of another, likely downstream protein called tau, which clumps within nerve cells in the form of tangles. In paragraph 9, accumulation of these proteins leads to nerve cell death, which leads to brain shrinkage, which we call atrophy, and which can be seen using MRI scans. The diagnosis of Alzheimer's dementia remains predominantly clinical, requiring evidence of decline over time in at least two cognitive areas such as memory, language, attention, or problem solving. Contemporary criteria also involve investigations including MRI or CT brain scanning, and in some cases spinal fluid or positron emission tomography scans. PET scans can be used both to visualize and quantify abnormal protein deposition within the living brain. For Insight 46, we use a tracer that is injected into the body, enters the brain, and which highlights where any amyloid is accumulating, like we can see in the scans here. Alzheimer's disease is only one of the many forms of dementia. Other causes include other neurodegenerative disorders due to accumulation of different proteins, and cerebrovascular disease where the blood supply to the brain is interrupted, for example from blood vessel narrowing, blockage, or bleeding. We go to the beginning of Insight 46. While developing what was to become the mental aging research program for our team, we acknowledged that it would be an exciting new direction to scan the brains of NSHD participants. Previous work at the Dementia Research Center at the University College London Institute of Neurology has shown that MRI scans from patients with rare genetic forms of Alzheimer's disease showed excess brain shrinkage occurred before symptoms started. In addition, UCL had installed the UK's first scanner that allowed MRI and PET to be measured simultaneously, so they could ask you the advantage of incorporating these two together. As NSHD participants were approaching the age of 70 and still relatively healthy, this raised an exciting possibility. Our two teams could combine forces and combine the uniquely rich life course data with state-of-the-art scanning to explore the brain changes that occur before symptoms become apparent in a birth cohort, something that had never been done before. So this is a PET MRI scanner. What we have found so far: 1. Amyloid accumulation starts before symptoms. We found that around 18% of cognitively normal people from the cohort had amyloid PET scans like those seen in people with Alzheimer's disease, a finding that tallies with other studies in people around the world who don't have symptoms. These individuals also had slightly lower performance on sensitive tests of cognition and slightly increased rates of brain shrinkage. While the significance of the findings for amyloid frequency is unclear, and hence our protocols and consent processes mean that unlike some MRI findings, we do not give the results to participants, we think that these individuals are at a higher risk of developing cognitive impairment in the future, something we plan to look out for closely in the years to come. 2. This is the second finding. Child cognitive tests indicate brain function later in life. We found that cognition assessed in childhood predicted cognition around 60 years later. This is consistent with the earlier findings for the whole cohort, suggesting that some aspects of cognitive performance are stable over a lifetime. This matters because cognitive function is not just about the mind, it helps to shape everyday skills, supports quality of life, and ultimately predicts how long we live. However, the level of cognitive performance can be potentially improved. In the same report, Education and Occupation in Midlife predicted later cognition after taking account of childhood cognition scores. We had seen this in the whole cohort too, which counters an old argument, still sometimes made, that education is nothing more than a marker of IQ. In other words, level of education and type of occupation can positively affect cognitive performance in later life regardless of cognitive skills in early childhood. It also emphasizes that education does not just increase opportunities, but has a significant effect on brain health. The third, importance of early heart health checks. Some of the first publications from the Insight 46 show that high and rising blood pressures in those aged in their 40s, and in some cases their 30s, predicted smaller brain volume. There are several possible mechanisms for this, including microstructure damage from high blood pressure and a higher burden of small blood vessel damage in the brain. The latter is thought to be a marker of brain frailty, raising the risk of stroke, dementia, depression, impaired mobility, and death. Similar outcomes were seen in relation to heart health in general, using an index that includes blood pressure, use of antihypertensive medication, diabetes, smoking, and high body weight. Conversely, falling blood pressure in later life may in some cases be a marker of poor health. Similar findings may also apply to body weight. A follow-up analysis found that declining body weight in the 2 years before the scan predicted the presence of amyloid. These findings have significant implications for public health, suggesting that routine checking of heart health and blood pressure in particular may need to start much younger than is typically recommended, probably at or before the age of 40. Then the fourth, a blood test for Alzheimer's disease. Most experts will agree that when we have new drugs for Alzheimer's disease, they are likely to have maximum benefits if taken earlier in the disease, and preventing the onset of cognitive decline would clearly be preferred to trying to slow or halt memory decline that has already started. It is unlikely that the expensive PET scans we are conducting in Insight 46 will be able to screen whole populations, so there is much interest in developing blood tests instead. Using state-of-the-art methods sensitive enough to detect 1 gram of salt dissolved in 1 million trillion liters of water, we were able to show that a blood marker is capable of detecting amyloid in the brain with about 85% accuracy. We are currently looking at a range of new blood tests that seem to be even better at detecting amyloid and at even lower cost. The prospect of new drugs that can clear amyloid from the brain provide even more reason to intensify efforts to identify amyloid pathology early, cheaply, and at scale. Studies using the whole NSHD cohort have also shown complex relationship between cognitive function and several bodily functions, including those of the lung, bone, and kidneys. This probably reflects biology shared between the brain and these organs. We are currently looking to see how these findings relate to the brain health measures we have made in Insight 46. A similar common cause story applies to depression and cognitive function, and we are currently looking into how depression relates to the brain. On the other hand, health-related behaviors such as smoking, physical exercise, and healthy diet genuinely seem to predict cognitive function, negatively for smoking, positively for exercise and diet. We have been emphasizing prediction of health problems, but it's equally important to understand resilience. Why can some people navigate through or escape these problems altogether even though they are at risk from genes or certain disadvantages in life? Does it come down to pure luck? But luck is of course just another way of saying we don't know something. When we go to the second article, this is about anti-amyloid pathway in Alzheimer's treatment. So they begin with new Alzheimer's drugs don't deserve the hype. Here is why. A prominent childhood memory is of my grandparents living with and then dying from dementia. As is universal with dementia, there was a double blow watching my grandparents lose their identity and seeing the suffering of those closest to them. As a junior doctor on a specialist dementia ward, when I was in my 20s, I watched the same stories play out for family after family, feeling largely powerless to help. Now in my 30s, I conduct public health research to understand what we can do to prevent, delay, or improve the experience of dementia, the leading cause of death in England. Naturally, this makes me desperate for good news on treatment options for Alzheimer's disease, the main cause of dementia. Enter 3 drugs, aducanumab, lecanemab, and donanemab, that remove amyloid, the protein thought to cause Alzheimer's disease. Unlike their many predecessors that also successfully removed amyloid from the brain, these drugs were the first to slow cognitive decline. Drugs with an ending of MAB are whole monoclonal antibodies. This breakthrough was hailed as the beginning of the end for Alzheimer's disease, but how useful are these drugs going to be? There are 4 key shortcomings to consider. Number 1 is the tiny benefit. In the donanemab trial, the people taking the drug declined on average by 10 points on a 144-point cognitive scale. The placebo group declined by 13 points. Consistent with the patterns in the trial of the other 2 drugs, this tells us that all groups in all these trials declined, and the amount of decline that was avoided by taking the drug in the case of donanemab, 3 points, was a lot smaller than the amount of decline that still occurred, 10 points. The difference in the amount of decline was so small that it would probably not be noticeable to the doctors looking after these patients. So the second is side effects. Through regular magnetic resonance imaging scans, 1 in 6 people taking lecanemab was found to have evidence of brain bleeding, and 1 in 8 had brain swelling. Regular scans will sometimes pick up these pathologies in dementia patients, and indeed 1 in 11 of those in the placebo group had evidence of bleeding, while 1 in 59 had swelling. For most people, these events were only detectable by MRI and not through showing any specific symptoms. However, the effects of these drugs' damage to the brain, particularly the long-term effects, are unknown. Sadly, there have also been a few deaths attributed to these drugs. Number 3, of course, they are very expensive. Aducanumab was marketed in the US at $45,000 per patient per year. Later, it was decreased to $20,000 due to demand. Lecanemab for $26,500. This is just for the drug itself. Health systems also need to pay for additional scans to test for eligibility, monitoring and managing of side effects, and staff to run infusion clinics. The donanemab trial suggested that treatments could end when the brain scan showed sufficient amyloid clearance, but we don't know if amyloid will return after some time. Regular monitoring for amyloid recurrence and repeated bouts of treatment would add further costs. There are other impositions for patients: attending centers every 2 to 4 weeks for drug infusions and regular monitoring, and worrying about the side effects. Number 4, highly selective trials. It's accepted that not all trial efficacy, the effects seen in a specialized trial context designed to maximize the likelihood of treatments working, such as including only uncomplicated cases, will convert into clinical effectiveness. Effectiveness is the effect seen when drugs are given to relatively more complex patients in busy real-world clinical settings. This is concerning because there is little wiggle room before the effects become undetectable. And while this is the case for all diseases, Alzheimer's is likely to be an extreme example. For every 10 patients that doctors thought might be eligible for these trials, 7 or 8 were rejected. People with brain pathologies other than amyloid, such as vascular damage or Lewy bodies, and those with significant other medical problems might have clouded the trial result and increased the risk of side effects, were excluded. If the drug eligibility is restricted to match the trial eligibility, then very few people will be eligible. If eligibility is broader, then already smaller effects are likely to be even smaller and side effects more pronounced. Profound shortcomings. There is more. Trials selected people at the earliest stages of the disease, that is, when symptoms had only recently developed, and successfully cleared amyloid, yet patients still declined almost as fast. So inevitably, researchers ask, maybe we need to start the drugs even earlier, but how? People in the trial were on average 5 to 10 years younger than most people are at Alzheimer's diagnosis in the US and UK. Catching people earlier in the disease is problematic because most people with amyloid but no cognitive symptoms won't get dementia before they die. Sadly, I don't think that drugs can make a big difference for people currently or soon to be living with Alzheimer's disease. Also, the shortcomings are so profound, despite decades of expensive trials and patient sacrifice. I think it's time to take the amyloid blinkers off and prioritize exploring other neglected options for treating dementia. This isn't the beginning of the end of Alzheimer's, but perhaps it should be the end of the anti-amyloid drug pathways. These are not possible questions, they are possible topics, so topics I'm emphasizing on that you guys can revise in preparation for any questions from these articles. The first one, and I think there is a very high chance that questions are going to come from here, are techniques that are used to image the brain. Number 1, they can ask about MRI, fMRI, PET, as well as CT scans. They'll ask you about the advantages and disadvantages, as well as how these techniques are carried out. So the descriptions, as well as advantages and disadvantages. Then we go to neurodegeneration. Of course, in your textbooks, you studied about Parkinson's disease, as well as depression. So they'll ask you about Parkinson's disease, dopamine, because remember, there is death of dopamine producing neurons in the brain. And so there is a high chance a question could come about Parkinson's disease and how dopamine is going to be maybe insufficient in the brain and how we can treat that using the L-dopa, which is a precursor to producing dopamine or for making dopamine. They will ask you about serotonin and depression. So, and then when we go to the nervous system here, they're going to ask you maybe how the nervous system works. They could ask questions about nerve impulse transmission, myelination and demyelination. I'm talking about axons that have myelin and those that have no myelin, and how this is going to affect the impulse transmission through those axons. What you need to remember here is axons with myelin are going to transfer or transmit the impulses really faster in comparison to those that do not have myelin. The reason for that is those that have myelin, there is going to be conduction only at the nodes of Ranvier, while the others, there'll be conduction all over the whole axon. Then they could ask questions relating to the axon diameter and impulse transmission, sympathetic versus parasympathetic nervous system, impulse transmission across the synapse, action potential generation, questions about nerve impulse transmission and muscle contraction. Here you could talk about the neuromuscular junction and how the impulses are going to trigger the release of calcium ions, Then they could talk about reflex actions. Remember the spinal reflex as well as the pupil reflex. They could include the eye. Again, still, since we are talking about neurons, they could connect to the eye, then go to rods and cones, rhodopsin, generator potential, and so many more. Another thing is questions about effects of drugs in the brain. Remember, most of the time it's at the synapse, whereby they could trigger an action potential or prevent generation of an action potential in the next neuron. Then about beta amyloid proteins, here they could ask you about protein synthesis, how proteins are going to be packaged, how proteins can leave some cells by exocytosis. Remember, these proteins are made in some neurons and they're going to aggregate outside these neurons or within the neurons, so they could talk about exocytosis. Then inflammation and inflammatory response. There is interaction between different proteins to form tangles as well as plaques. So here they could ask questions about the intermolecular forces that form between the different proteins as they come together to produce aggregates of proteins. Here remember about the hydrophobic and hydrophilic links and how these can come into play as different protein molecules interact with each other or as they interact with other surfaces. Then we can go to cardiovascular risk factors like smoking and inactivity and how these could affect the brain. Remember, again, I'm not saying that they will bring about atherosclerosis and all that, but remember some of these cardiovascular risk factors lead to formation of plaques within blood vessels, and this could lead to the narrowing, which could later lead to complications like formation of blood clots, and if these occur in a region of the brain, they could lead to these complications whereby brain activity is going to be affected, like in cases where we see strokes and so many more. Next, I want to go to involvement of genetics. This could be about how you can be able to analyze certain genes to know if somebody is at risk of developing dementia. For example, which genes have been expressed. Here you could use microarray technique and other techniques that are used for detecting expressed genes. They could bring in questions about how control of gene expression can occur, and then using genetic modification as a potential solution of dealing with some of these problems that lead to neurodegeneration and therefore the different types of dementia. Then questions to do with the nervous system and the effect on other systems, like how problems in the nervous system can lead to problems with the muscular contraction, how those can lead to problems within the heart, and how those could lead to problems within the ventilation system or in how the lungs work. Extra topics that I think they could talk about may be clinical trials. We've seen from the article they talked about clinical trials, so there is a possibility that they could bring about the 3-phase clinical trial and so many more. Then about kidneys, and again I'm talking about this being something that could be a small question, but they could ask about questions relating to the kidneys and how chemicals like hormones affect cellular function. Remember, these hormones act as transcription factors, or they can produce a second messenger that acts as a transcription factor and leads to expression of certain genes. So maybe this could be chemicals produced in some other cells or produced by the bodies of those people that act as transcription factors, affecting which genes are going to be transcribed and therefore leading to the formation of these proteins that tangle up and cause neurodegeneration and the different forms of dementia. So these are the possible topics I'm focusing on. And that brings us to the end of this article. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.